Okay, everyone, when we left off with part 1 of chapter 13, we had talked about the process of meiosis and mitosis. So let's just review and compare these two one more time, with mitosis being on the left and meiosis being on the right. Now, in both cases, they start out with a diploid parent cell, meaning that there are two copies of each chromosome. Now, in both of these first diagrams, this is from your book, and what they really mean here is that both of these cells are in interphase. Now, here's what I don't love about the diagram. You guys should know from my lectures that these drawings of chromosomes during interphase are a little bit misleading, because what we know is that the DNA is actually in the form of chromatin at this point. And that it's relaxed, right? It's not really formed into these compact structures. So what this really should look like is a bunch of blue spaghetti here and a bunch of red spaghetti here. But I think for ease of visualization, they've drawn it the way they have. Now, you'll remember that interphase for both of these guys consists of the first gap, which is when growth and functioning occurs, the S phase, which is when DNA synthesis occurs, and then the second gap, which is when the cell prepares to divide. That is the same for both of these. They both have a G1, an S, and a G2, okay? And you notice here it says chromosome replication. That's gonna happen during the S phase, whether we are talking about mitosis or meiosis. Now, let's look at what happens during prophase in mitosis, okay? So we're just gonna go on this side. During prophase, our chromosomes are gonna condense, the nuclear envelope is gonna break down. They also don't mention, but the spindle apparatus is going to form. Okay. Let's contrast that with prophase 1, because remember, we're going to do this twice in meiosis. Chromosomes condense, that's the same. Nuclear envelope breaks down, that's the same. Now here's some differences. Bivalents form, remember, bivalents are homologous pairs. I'm going to write homologs. And the process by which they match up is called synapsis. Okay, so these bivalents is another way of saying homologs that are matched up together. And the process when they match up is synapsis. The other thing is crossing over is going to occur here. We're going to talk about that in just a second. Chiasmata is a term that refers to crossing over. So you can just write crossing over here. And what crossing over is the swapping of material from one homologous chromosome to the other. So I don't know if you guys can make it out, but on here, you may want to blow this up later on your PowerPoint. You can see that one of the red chromatids has overlapped one of the blue, okay? And you can actually see it a little bit better here, but it happens during prophase, okay? Now, let's get to metaphase. If we are talking about mitosis, the thing that you will note is that these folks have lined up single file, okay? Here in metaphase 1 of meiosis, they are 2 by 2, right? With this being a homologous pair. Okay. And then this diagram skips anaphase and telophase, but you know that what happens in mitosis is that we're going to have spindle fibers that pull that way, and that way, that way, and that way, resulting in sister chromatids separating and the nuclear envelope reforming. At the end of this process, we will have two, actually let's write that out with a T, because that's how I remember it. We will have 2 exact copies. There's a T, and guess what? Mitosis has a T. Mitosis, 2 exact copies, okay? At the end of meiosis 1, when we go through anaphase and telophase, what happens here is we're going to have spindle fibers pull this entire structure in this direction. And we're going to have spindle fibers, also known as microtubules, pull this entire structure in that direction, so that at the very end of meiosis 1, you now have gone from being a diploid cell, which incidentally, diploid cells are often designated by the shorthand 2N, to a haploid cell, which is designated by the shorthand N. So we've gone from having 2 copies of each chromosome to 1 copy of each chromosome. Now, don't be confused, because this is 1 chromosome, and we know that because we have 1 centromere. But this is essentially like 2 pieces of paper that are stapled together. So these are actually 2 copies of the DNA, but it's 1 chromosome. I hope that makes sense to you. Same with this one. We have 2 copies of the DNA, but 1 chromosome. All right, what's going to happen in meiosis 2 is that we're going to pull these apart. We're going to pull these apart, and you can see that the result of meiosis totally is 4 haploid, and I'm going to shorthand that as N, 4 haploid cells. Okay, contrast that with mitosis, where you have 2 diploid, here's our shorthand, cells. All right, now you may be asking yourself, why in the world would we go through this whole process, right? This is fairly complex. And the answer really has to do with sexual reproduction, right? I already told you guys at the beginning of the part 1 lecture that the issue is, if you are a diploid organism, which we are, so let's use humans as an example. If we took 2 normal diploid human cells that have 46 chromosomes each, 1 from mom, so I'll put mom here, and 1 from dad. If we took 2 diploid cells and we fused them, then suddenly we would have 92 chromosomes, right? And if you went through this process every time, you would have increasing numbers of chromosomes every time organisms reproduced. So the reason that we undergo meiosis is so that we can have sexual reproduction. Now, let's define sexual reproduction. Sexual reproduction is the fusion of gametes, so it's down here. In other words, sexual reproduction involves fertilization. Let's contrast that with asexual reproduction. Asexual reproduction is just simple cell division, and it produces clones. Clones are exact copies. Have we learned any cellular processes that produce exact copies? Well, of course we have. Asexual reproduction occurs via mitosis, okay? And you find this in plants. You find this in some unicellular organisms like yeast, okay? Whereas sexual reproduction you find also in some plants, a lot of plants actually, and you find it in animals, okay? Now here's the big question. Asexual reproduction seems pretty simple. You just undergo mitosis. You're just undergoing 1 cell division. So why on earth would sexual reproduction even exist? Okay, the answer has to do with this slide. Sexual reproduction exists because it generates genetic variation, okay? Because you can have, let's see here. Here we have a sperm that is fusing with an egg, right? So you've got a mom and a dad. So I guess I have that backwards, don't I? You've got a dad and a mom, okay? This dad has a certain set of genes and this mom has a certain set of genes. And so when you add these two together, you get a unique and new combination. If this dad were to later mate with a different mom, it would be a whole new combination of genes, wouldn't it? And so what sexual reproduction does is it produces unique organisms through genetic variation. Now, why would this be important? Think to yourself, why would a population want to have a lot of different organisms with different genetic makeup? Well, it's actually kind of timely that we talk about this, because the example that we often give is that we want populations to be genetically diverse and have genetic variation, because in the event that a disease comes along, you want the disease might wipe out some of your population, but you want to have the chance that at least some individuals, through genetic variation, have the ability to survive that disease. COVID-19 is a great example. Some of us are really susceptible to COVID-19, and there's been 200,000 deaths in this country. Some of us get COVID-19, but we exhibit very little symptoms and we recover quite quickly. So that's a great example of genetic variation. If COVID-19 came along and everyone was genetically the same, it could wipe out our entire species. But because we have all these differences, some of us do quite well and survive. So meiosis's big importance is that it is a source of genetic variation. And I want to tell you about 3 different sources of genetic variation. The first one is simply, how can I put this? Mate choice. And what I mean by that is if Beyonce and Jay-Z have a baby, that's a great baby, right? But then if Beyonce were to suddenly have a baby with Nicki Minaj, that's going to be a whole different child, isn't it? It's going to have a whole different genetic makeup. So depending upon who is partnering with whom, that will give you your very first set of genetic variation, okay? The Jay-Z Nicki Minaj baby is going to have a different set of genes than the Jay-Z Beyonce baby, right? Okay, let's look at another source of genetic variation that we get just out of the process of meiosis, okay? And this source of genetic variation has to do with mom and dad, but it's a little bit different, okay? This is called independent assortment. Now, I want you to think about your mom and dad, and I want you to think about maybe someone has told you before that you have your mom's eyes and maybe you have your dad's big feet, okay? So you get some of your genes from mom and some of your genes from dad. Well, that's kind of related to this idea of independent assortment. Let's say that you are developing gametes, whether you're male or female, maybe you're developing an egg or maybe you're developing a sperm, okay? You've got this set of chromosomes from mom here in the red, and you've got this set of chromosomes from dad here in the blue, okay? When you get ready to have your own offspring and you're developing gametes, you may end up with a situation where, let's say that if the blue lines up on this side, blue, blue, and the red lines up on this side, red, red, then you could end up with a situation where you end up, see, we have 2 blue chromosomes, 2 blue chromosomes, 2 red chromosomes, 2 red chromosomes. So this egg cell is going to have nothing but paternal genes. Oops, why did this stop? There we go. Nothing but paternal genes. Whereas this egg cell would have nothing but maternal genes. Okay. Here, we can see that they've lined up differently. You have a paternal chromosome here and a maternal here, but it's backwards here, isn't it? So when these segregate out, you can see you've got 1 dad chromosome and 1 mom chromosome, but they're opposite in this set, aren't they? So this is a unique, even though it came from the same stock here. If you look, we have really the same set of chromosomes here and here, but they've lined up differently, haven't they? Okay, red's on the left, or sorry, blue's on the left, red's on the right, and on this one, it's the opposite. What if we did this 23 different times? What if we had, 23 different times where these guys lined up differently? What are all these possible combinations? Well, the way to work that out mathematically is it is 2 to the 23rd. You have 2 chromosomes, one from mom and one from dad. There's 23 different ways they can line up on the left or the right. That's a lot of different eggs or sperm cells that you can make just based on how these things line up. So independent assortment is whether maternal or paternal chromosomes end up in a gamete, and it's random. There's no process that determines which side the red's gonna be on or which side the blue's gonna be on. So this is the number 2 way we get genetic variation out of meiosis. Number 1 way was mate choice, just different mates mating with each other, or different sperm from different individuals fertilizing different eggs from different individuals. Number 2 has to do with even within the same 2 individuals. Like my husband and I have 3 children. Those 3 children look quite different from one another because of this right here, this idea of independent assortment, where each of my egg cells was a little different from the other because my chromosomes lined up differently during meiosis, okay? The third way that we can get genetic variation out of meiosis is through a process called crossing over. And I've mentioned this a couple of times. It occurs during prophase 1, prophase 1 of meiosis 1, right? Prophase 1, meiosis 1. Anytime you see a 1 after a phase, that means it occurs in meiosis 1, all right? So what happens there is, remember that we had this process called synapsis, where the chromosomes partner up. They find their homologous pairs. And what will sometimes happen is the chromatid of one chromosome will tangle up with the chromatid of the other, and they'll actually swap genetic information. So now this is a new unique combination. You can see here that all of these were dad's genes, but now we have a chromosome that's mostly dad and partly mom. And on this side, we have a chromosome that's mostly mom and partly dad. So this is now a unique combination as well. So if you add these 3 features together, if you add up the fact that you can have different individuals mating, different mates, and if you add the fact that for each mate, their eggs and sperm are the result of independent assortment, in other words, random placing of maternal and paternal chromosomes, and then finally, if you add in this idea of crossing over, holy cow, you can get a ton of genetic variation. And that's very good for the population, because if environmental conditions change, sorry, my stylus is crazy tonight, variation. If environmental conditions change, you want to have lots and lots of different diversity within your population so that some individuals will do well in the face of environmental changes, okay? All right, let's move on from there. Really quickly, this is another close-up of crossing over just to help you visualize it. So again, this is prophase 1. And you can see that what has happened here is, as these guys find one another, Here it looks like these guys might swap some material, and it looks like the guys might swap some material. Now this area where these guys overlap, where their chromatids become entangled and they cross one another, that area is called a chiasmata. So if we look over here, can you see where you have like an X? Chi, this right here, chi is the Greek letter X. And so this word chiasmata comes from the fact that where these chromatids cross over, it looks like a letter X. So each of these little letter X's are chiasmata, okay? Here, I wanted to point out for you that we have a summary of the differences between meiosis and mitosis, okay? But the big deal that I want you to remember about meiosis, and the reason that it exists, even though it is more complex, it exists because sexual reproduction, which is what meiosis is, or what meiosis helps to enable, sexual reproduction is, it generates genetic variation, okay? Now, I will tell you that I really like this summary table that compares and contrasts meiosis and mitosis. So I would spend some time with this, preparing for your quiz and preparing for your exam in a couple weeks, okay? Now, meiosis is a fairly complex process. And so sometimes mistakes happen during meiosis. And you've probably actually heard of some of these mistakes, although you may not have realized that it was meiosis causing them. So let's take a look at a really common mistake that can happen during meiosis that you would have heard of, okay? And this is a phenomenon called non-disjunction. This name is going to make tons of sense to you once you see what happens. So we are starting out with a cell here that is 2N, okay? 2N, meaning it is diploid. So we have 2 copies, 2 sets of homologs here, right? Now, what has happened in this mistaken meiosis 1 is that a spindle fiber has grabbed this guy from this side. It reached over and grabbed this guy from this side. Well, that's going to result in an error because normally you'd have spindle fibers attaching here to those on this side, and you'd have these from this side attaching here. That is not what happened. Let's review that. So under normal circumstances, these spindle fibers would attach to these guys on this side. These spindle fibers would attach to these guys on this side. Instead, what happened in this scenario, a mistake was made. So these attached properly, and then there was a spindle fiber that reached over and grabbed this one. This one attached normally. Now, when these start to pull back, when these start to go back toward the opposite poles of the cell, you can imagine what's going to happen, and we have it pictured here. 3 of these guys end up going in that direction, and this one ends up going in that direction. That means that at the end of meiosis 1, we have a problem. This cell here has an extra chromosome, and this cell here is missing a chromosome, right? Because if we look at the original cell, we have 2 homologous pairs, and if we reduce it down, we should have 2 chromosomes in each cell, right? But we don't. So if we undergo meiosis 2, which is what's happened here, you have these that have 3 chromosomes, which they have designated as haploid plus 1, haploid plus 1. These, which only have 1 chromosome, which is haploid minus 1 and haploid minus 1. This scenario here, where you're missing, this is going to be if this becomes like the egg or something, this will often end up in a non-viable offspring. Sometimes this will also result in a non-viable offspring, but there's a condition that you're probably familiar with that does result from this problem. And that condition is called trisomy 21. Trisomy 21 is also known as Down syndrome. It is characterized by cognitive dysfunction. So in other words, these are usually individuals with lower IQs. They have a very distinctive set of facial features. They often have a flattened nose and kind of like really thin eyes. They can have kind of a stocky body stature. Now, how did this arise? Well, what happened here is we had a situation where for chromosome 21, maybe let's say dad, oops, let me erase that, dad donated 1 copy of chromosome 21, and then let's draw an egg cell for mom. I'm gonna do that in red. But through non-disjunction, mom's egg cell ended up with 2 copies. And so when these fused together, you ended up with a baby that had 1, 2, 3 copies of chromosome 21. Let's go back and look at how that could happen via non-disjunction. Excuse me. Can y'all make out here that this individual, can you see that, like, let me back up. When this egg was made, can you see that these 2 homologs here ended up in this cell? So this cell has 1 copy of this chromosome. Let me change colors so you can see that a little better. This egg cell has 1 copy of this long chromosome, and it's got 2 copies of this short chromosome. So what if these were chromosome 21? Well, you'd end up with this situation here, okay? So this is an example of non-disjunction. Okay, and non-disjunction is what we call a trisomy. It's called trisomy 21. And it makes sense, right? 1, 2, 3, trisomy, 3 chromosomes, okay? Anytime you have a situation, there's multiple other diseases that can result from problems with meiosis, and you can end up with abnormal chromosomes. Anytime you have a variation from the normal 2N, which is what you see here and here and here, that there's a name for that, and it's called aneuploidy. Okay, aneuploidy is just when you have some deviation from the normal 2N number of chromosomes, whether you're missing one or whether you have extra, okay? Now, what we know is that these errors in meiosis tend to increase, particularly if we have a mother of advanced age, okay? So these mistakes go up. Look at this graph. We have starting with age 30 here and ending with age 50 here. You have the incidence of Down syndrome per number of births. So if you were to have a child at age 30, you would see that there's about, if you go through the population, about 1 in 900 births to women that are age 30 are Down syndrome babies. But if you take this all the way out to like 48 or 49-year-old, I guess, is that supposed to be 50? Looks to me like it's about 49. To about 49-year-old mom, what you find is that 1 in 10 births results in Down syndrome. Why is that? Well, it has to do with meiosis messing up. So why do things go wrong? Well, I showed you that microtubule attachment to chromosomes in meiosis 1. Remember how that microtubule grabbed the wrong chromosome? You can have problems when you separate crossing over. Sometimes that can create breaks in chromosomes. You can have premature separation of sister chromatids. All of these are errors that can happen during meiosis. And the reason that these errors increase as mothers age is in humans, this is really unique to humans, it's because our eggs, they're all formed when you're an embryo, when you're in utero, okay? So when you're born, formed in utero. So if you are biologically female, when you're born, you have all of the egg cells that you're ever going to have for the rest of your life. Now, those egg cells are immature. They're actually sitting in a state of arrested development, and that state is in prophase 1. And then what happens each month as you ovulate is these eggs that are in prophase 1, one or two of them will go through meiosis to become mature egg cells, okay? So that means that if you are 48 or 50 years old, your eggs are also 48 or 50 years old. That means that these features, the microtubules, your chromosomes, all of these features may have degraded over time so that increases the opportunity for mistakes to happen. Now, we don't see this in males because they consistently create sperm all throughout their life, all right? So, let's see, I think that's everything that I wanted to tell you about chapter 13. We are not going to go through the process of egg and sperm formation in this chapter. That's something that you will take if you take genetics with me or other courses in the future, but I'm happy to answer questions about it if you have questions during class time. This is going to be the end of the material that you will have on your quiz on Friday. So good luck, and as always, I'm happy to answer any questions. Bye-bye.